Hi, everyone. So you have finally made it to chapter 7. This is the last chapter that will be included on your exam 1 if you are watching this video in the fall of 2020. Remember, exam 1 is going to take place on September 4th during your scheduled class time. So you can't take it before or after. Remember that the exam will show up on Blackboard at the beginning of your class period. You will have the duration of the class period to take the exam, which is 50 minutes. Now, remember that we did not end up using the Respondus browser for your quiz. So you can use your regular internet browser, okay? However, my intention is that you answer these exam questions using your knowledge as opposed to the knowledge that you can seek on the internet. Obviously, I have no way of checking whether you are looking things up on the internet. So in order to sort of discourage you, I know it's tempting, but to discourage you from using the internet to look things up, a couple of mechanisms that I will be using. You'll have 50 minutes to complete this exam, but there will be more than 50 questions. Some of them will be very simple questions. Some of them will be much harder. I will also be disabling your ability to backtrack, just like on the quiz. So you have to answer the question you're on and move forward. And the reason that I do that is because I know that you'll have to budget your time if I use those two practices. If you spend a whole bunch of time looking up the answer to question 5, you don't know what I might ask on question 25, and you may very well run out of time to complete the exam. So my advice to you is do not think that you're going to be able to use the internet and make your way through this exam without studying. You're going to have to use your brain to answer these questions with the understanding you've gained from these videos. Okay? All right, folks. So let's talk a little bit about this chapter. Up until now, we've been kind of going over all the things that might make up the component parts of a cell, right? We've been talking about proteins, nucleic acids, carbohydrates, and then finally lipids, which made up the cell membrane, right? Now we've finally arrived at a point where we're going to put all those things together into the functioning unit of life that we call the cell. Remember that the cell theory tells us 3 things that are really important about cells. The first one is that all of the organisms, I'm going to put all orgs, are made up of 1 or more cells, right? So after we invented microscopes, we started looking at all the things we could under the microscope, and what we found is that all the living things that we could examine, they were all made up of at least 1 or more cells. The other thing that we noticed is that cells are the basic unit of life. So I'm going to put cells equal life. What do I mean by that? I mean that if you took apart a cell and you looked at just, say, the nucleus by itself, or just a ribosome by itself, those entities alone are not alive. You have to have all of the components of the cell together in order to have all of the attributes that we consider for living things to be alive. And then the last thing that cell theory says is that all cells come from other cells, right? From pre-existing or other cells. So be sure that you remember all 3 parts of the cell theory. Now, in thinking about where we're headed in this chapter, I kind of think of this chapter as occurring in 2 parts. This first chapter is really what I consider a review of stuff that you should have had in high school and middle school science, although I'll give you a little bit more detail. But what we're primarily going to do is go over the parts of the prokaryotic and the eukaryotic cell. And these occur in sections 1 and 2 of chapter 7, and they are information that will be included on your first exam, exam 1. Again, this only applies to the fall of 2020. This stuff down here, we're going to start looking at what we call cell systems, a whole bunch of different parts of the cell that work together to carry out some functions. And this information is going to be what's included on exam 2. Okay? So again, if you're watching this fall of 2020, you really are only going to need to cover this stuff for your exam, and that's what I'll be going over in this video lecture. All right? Now, most of you know that cells kind of come in two flavors, right? You can think of them as being prokaryotic or eukaryotic. And if I were to ask most of you right this second what the primary difference is, you would probably talk to me about the presence or absence of a nucleus. Most students know that a eukaryotic cell has a nucleus while a prokaryotic cell doesn't. We often think of prokaryotic cells as being a little bit simpler in structure than eukaryotes. Now, where are we going to find prokaryotic cells versus eukaryotic cells? Remember that there are 3 domains of life. I think I briefly touched on this in chapter 1. So here's one domain up here in the purple, the second domain here, and the third domain here, okay? This first domain contains bacteria, okay? It's written for you right here. And these are all different groups of bacteria that you will study if you go on to take microbiology. The second domain is one that we didn't even talk about when I was a high school student, or really even when I was an undergrad, because they've only recently begun to be studied in earnest, I would say in the last 20 years or so. And so these are the archaea, or archaea, depending on how you say that. I have a good friend that studies these guys, and she probably would tell me that whatever I'm saying is wrong, but I don't care how you say it. So the archaea. So what's their deal? Well, a lot of what I'm going to tell you today about prokaryotes applies primarily to bacteria because that's what we've studied the best. It turns out that archaea, their cells are slightly different than those of bacteria. They are still considered prokaryotes because they do lack a nucleus and they lack membrane-bound organelles, but much of the chemistry of archaeal cells is quite different than that of bacterial cells, okay? The third, so let me back up. So both of these, 1 and 2, are both going to be prokaryotic cells. And these are unicellular organisms that, again, lack nuclei. These folks down here in domain eukarya, these are our eukaryotes. Makes sense. Eukarya, eukaryotes, right? These contain things like fungi, animals, and plants, okay? So hopefully all of that is a bit of review for you. Now, before we jump into talking about what prokaryotes look like on the inside and what eukaryotes look like on the inside, let's kind of take a step back and think about how we got here. Up till now, we've been studying sort of individual molecules. So let's kind of think about how we got to the cell from individual molecules. And to get here, we don't really know exactly how the first cells evolved, but we can come up with some hypotheses. So I want you to think back to chapter 1. And in chapter 1, I told you a story about the Urey-Miller experiment. I think your book might just call it the Miller experiment. And so just a quick refresher, you should know this for your exam, but in this experiment, they were kind of questioning, where did some of these macromolecules for life come from? And so they set up a situation where they had water that they boiled and it became water vapor. They had some very simple molecules. This is methane, this is ammonia, water and hydrogen gas. And they also set up a simulated lightning. So water vapor, simple molecules, and lightning simulate what we think the early Earth conditions looked like. And they let this experiment run for a while. And what they would do is, as the water vapor would move through this chamber and be exposed to energy, and you'd have all these simple molecules in here, then they would let the water vapor cool down and they would collect some of that cooled solution. And what they found is simple organic molecules occurred after a few days, things like amino acids. And you could imagine that over time you might end up with things like nucleotides, okay? So once we have some organic molecules, the next thing that could have happened refers to something we talked about in chapter 4. In chapter 4, I told you about something called a ribozyme. And just to refresh your memory, A ribozyme was a single strand of RNA that kind of folded up on itself in a tertiary structure like a knot. And so in this way, it looked a little bit like an enzyme, only instead of being made of proteins, this is made of RNA. And one particular ribozyme that was interesting was able to do this very neat trick where it could take some RNA nucleotides, some ribonucleotides, so there's my phosphate, and there's my base. And it could link up the sugar phosphate backbone to make more copies of RNA. So it was kind of a self-replicating machine. It could take individual nucleotides, link them together, and make copies of RNA. In so doing that, what we see here are sort of the emergence of the first properties of life. Remember that life had 5 attributes, right? We now have the ability to have information in the form of an RNA template. We now have the ability to replicate. And we actually have the ability to evolve. Because if you remember me talking a little bit from that chapter 4 lecture, and you can go back and study that in your book if it didn't make sense. But if we took an RNA template and we copy it, but we make a mistake in copying it, maybe we take this base and it's supposed to be an A and we accidentally insert a C, we've gotten a mutation that may or may not end up propagating and causing this RNA to evolve. So now we have 3 of the 5 attributes necessary for life. Now, the next step, so we've got this plus this, the next possible thing that could have happened has to do with what I told you in chapter 6. And in chapter 6, I told you that lipid bilayers, they spontaneously form, I can't talk, lipid bilayers form spheres. They spontaneously make these little bubble membranes, right? So here you can see I have a phospholipid. Just to review, you've got a polar head and 2 non-polar tails, right? These form linkages. And when you have a phospholipid bilayer and you put it in water, it spontaneously forms a sphere. Now, what if when the sphere formed, we were able to trap one of these ribozymes along with some individual nucleotides? You might have the rudimentary beginnings of a cell, right? So we don't know for sure how cells got here, but we do have some thoughts about some processes that might have led to the evolution of the first cells. All right. Now, while we don't know exactly how cells got here, we do know what they look like today. And as I mentioned, you can really divide them into 2 flavors, eukaryotes and prokaryotes, okay? So let's compare and contrast these 2. Now, these designations are primarily, let me get rid of this, primarily based on what we call cell morphology, which is basically the study of, an ology is study, morph means shape. So these designations are based on the cell shape and structure. Why do I say that? Well, I already told you that while we say that both bacteria, and I'm going to abbreviate that as BAC, and archaea are prokaryotes, they do have some pretty important chemical differences between them. But because they lack nuclei and because they lack membranes, we go ahead and call both of them prokaryotic cells. And so that designation is based on how they look, all right? So one of the first differences is the presence of nuclei, but let's go ahead and make that more generalized. Let's just say the presence of membrane-bound organelles. Now this term organelles, if that is a new vocabulary word for you, I'm going to encourage you to look that up. So things that are enveloped inside a membrane. So like a nucleus is enveloped inside a membrane, so that fits. If you had something like a Golgi body or a vacuole, and it's surrounded by a membrane, then that also fits. We do not see these in prokaryotes. Another big difference is their size, okay? So this, image that I have for you from your text is a little bit misleading because we've blown these up. These are micrographs, so pictures that are taken through a microscope, and we've blown them up so that you can see some of the interior structures. But that sort of leads to the deception that maybe they're the same size. So let me draw your attention here to the size bar. Can you see that this is 0.1 micrometers? That's how you say that. This is a micro symbol, and that's a meter symbol. This is 0.1 micrometers, and this is 1 micrometer. So if I were to write this out for you, this would be 0.12345 of a meter. And this one would be 0.1234567 of a meter. So this cell is 10 times bigger than this cell. And that is generally true of eukaryotic cells. They are much bigger than prokaryotic cells, and we'll talk about why. Eukaryotic cells can be somewhere typically, I'm sure there's some exceptions, but in general, you can think of them as being about 5 to 100 micrometers across, whereas the typical prokaryotic cell is going to be 1 to 10 micrometers across. Again, I'm sure that there are some exceptions that I'm unaware of. All right, so let's jump in and let's start out with what some would argue is the simplest of the two, which is the prokaryotic cell. And let's learn some of those parts. So the easiest way for us to do that is going to be for us to draw it out. I'm going to draw first and foremost a shape. And I mean for this shape to convey the cell membrane. So this right here, I'm just going to put MEM for membrane. Now I've drawn it as a single line, but I want you to remember that cell membranes are composed of lipid bilayers where you have a phosphoglycerol head and then a 2-lipid tail, okay? One of the big differences between archaea and bacteria is the chemistry of these lipid tails, but I don't expect you to know that. It's mentioned in your book, but you don't have to know that, okay? So even though I know that this is a phospholipid bilayer and I expect you to know that too, I'm gonna just draw it as a single line for purposes of keeping our diagram clean. So cell membrane, check. The next thing that you will find in a bacterial cell, so I'm gonna really draw this as a bacterial cell. Archaea, as I mentioned, are slightly different, but we really know a lot about bacteria. All right, what I'm drawing for you here is a cell wall, okay? And maybe you remember from our discussion in chapter 5, this cell wall is made up of a structural carbohydrate that's coupled with some proteins called peptidoglycan. Now, if you're a nursing student or a pre-med student, you'll probably go on to take microbiology, and you'll learn that in bacteria, the placement of this cell wall is important as to whether you're dealing with what we call a gram-positive or a gram-negative bacterium, okay? So like gram-positive bacteria tend to have thick cell walls on the outside of the cell, whereas gram-negative bacteria have like thinner cell walls and they're sandwiched between two membranes. I'm not going to ask you to know that for purposes of your Biology 105 class, but I just thought you might find that interesting if you're interested in a health-related career. Another thing that you might find interesting is that the way that many antibiotics work is by targeting the synthesis of this cell wall. Basically, most antibiotics inhibit that cell wall synthesis in bacteria, and that's how they shut down their replication and their living. So I thought you might find that interesting as well. All right, now, so we've got our cell membrane, we've got our cell wall. Check and check. All right, let's next label the cytoplasm. So the cytoplasm is sort of like all the guts of the cell, all right? So it's the fluid that's in here, as well as all of the stuff that's suspended in that fluid, all right? And one of the first things that we're going to find suspended in that fluid in a bacteria is a large circular chromosome, okay? And the region occupied by this large circular chromosome is called the nucleoid region. So we don't have a true nucleus, but we have a region that we call nucleoid, which kind of means nucleus-like, and that's the region where this chromosome is located. Now, just to review, and so that everyone's on the same page, what's a chromosome? Well, a chromosome is made up of DNA, and it contains the genes or the information for how to make more copies of this bacterium. It also contains all the information for making all the proteins that are needed for this bacteria to function. But it's not well, let me go backwards. Okay, before I go on, one interesting feature, so you may have been surprised to learn that there's only one chromosome in a prokaryotic cell or in a bacterial cell, and that it is circular. Because you probably know from previous classes that if you were to look into the nucleus of, say, a human cell, there would be multiple linear chromosomes. As a matter of fact, in a normal human body cell, you would find 46 linear chromosomes, 23 different types, okay? And we'll talk more about that later. So that's another difference between prokaryotes and eukaryotes. Prokaryotes have one chromosome, it's circular, and they tend to have less genetic material overall, okay? In addition to the chromosome, in some bacteria, you'll find a little accessory piece of DNA. It's a circular piece of DNA, and it doesn't contain all of the genes that are necessary for this bacteria to live. This is called a plasmid. It just contains like interesting additional information, right? So if you just had a plasmid and you didn't have the chromosome, this bacteria could not live, but it contains additional information. For example, you guys probably know some bacteria are antibiotic resistant. So the gene for antibiotic resistance is often contained on a plasmid. All right, so these are just sort of extra informational genes that you'll find in some bacteria. All right, the next thing I want to talk about that you will find inside of a prokaryotic cell are ribosomes. Now, ribosomes are interesting because they are a combination molecule. They're going to be made up of both RNA and proteins. I'm going to put a lot of them as little dots in here. So let me label it for you. Now, what a ribosome does is it takes the genetic information from the chromosome and it translates it into protein, okay? So it is responsible for what we call protein synthesis. And I'm going to teach you all about how that process occurs, okay? So ribosome's job is protein synthesis. It takes the DNA information and then it gets transcribed into RNA and then translated into protein. This is where translation occurs, okay? All right, so we've got that one, check. Another thing that you will find inside of these cells is, and it's kind of hard to see under normal microscopy, and it took us a while to understand that prokaryotic cells had this, but I'm trying to draw for you some like really thin fibers here. Okay, these are protein fibers that make up what we call the cytoskeleton. So the cytoskeleton sort of functions as scaffolding to give this thing structure, okay? So I'm going to draw that in. Okay, there's your cytoskeleton. You'll also find on many bacteria an accessory structure, again, made up of some protein filaments that protrudes. There might be one or more of these, but these function in movement. These are called flagella, plural, or flagellum, singular. So I'm only going to draw one. So I'm going to make that singular. Okay, this functions in movement. Okay, and then lastly, the last structure I want you to know is sometimes you'll have these needle-like structures that protrude off the outside of bacteria and they make it look kind of fuzzy, okay? These are called, and I'm not gonna write it out since we're right here, they're called fimbriae. And what they do is they help bacteria to grab onto surfaces, and sometimes they can grab onto each other, they can grab onto like rocks or something in the ocean or onto other surfaces and form biofilms is what we call that. So they can kind of coat surfaces by using these fimbriae to attach to a solid surface. All right, so that is our very basic diagram of a prokaryotic cell. Let's look at a better diagram, okay? So this is what I just drew for you, but in a much better way, okay? So here, I've got, look at the bar I mentioned in your notes here. Note the size. These things are pretty small, but let's look here. So we have ribosomes. Again, their job is protein synthesis. Here you have this large spaghetti noodle looking thing. If you were to trace it all around, what you would find is that it makes a circle. So it is a singular chromosome, okay? All of the guts of this cell constitute the cytoplasm. On the inside in here, okay, you have the plasma membrane, and on the outside, you have the cell wall, so that's the yellow stuff. Here is our accessory bit of circular DNA called the plasmid that has some extra genes, all right? And the only thing that I think we talked about that's missing here are our little projections off the outside, like our flagellum or our fimbriae, okay? So let's zoom in a little bit more on some of these structures. So I wanted to show you that back up in here, I drew the chromosome fairly small and made it look like it's not that big, right? But actually, I wanted you to know that even though it is a single chromosome, it's actually, in order for it to fit inside the cell, it is packaged into these circles through a process called supercoiling. So imagine that you took a rubber band and then you twisted it up on itself, right? So it would be all twisty, 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 twisty. Okay, I'm not doing a very good job of drawing it out. And if you twisted it up, what you would do is you would be condensing it, right? To make it smaller, to make it take up less room. And that's exactly what happens with bacterial chromosomes. They are supercoiled. This is a form of tertiary structure of DNA. So that should take you back to chapter 4. And if that doesn't make sense to you, you better go back and review that. If we unwound this and uncoiled it, this is a single E. coli bacterium, and this is all of its unwound DNA. Do you see how much room this would take up if we didn't twist it up into these tight little supercoils, okay? So just make sure that you understand that while there's one chromosome, it still takes up some space. And so for it to get packaged, we twist it up to condense it down. Another thing I wanted to show you is some of these external structures. So I mentioned a flagellum, which is what this is right now. And that is used for helping this thing move around. These are those fimbriae, and that is used to help these guys adhere to surfaces, okay? Now, I also wanted to mention that there are some bacteria and some archaea that are capable of photosynthesizing, all right? And we're actually going to talk about that a little bit more when we talk about plant cells. In those prokaryotic cells, what you'll find is a whole bunch of additional membranes. There are proteins embedded inside these membranes that carry out the process of photosynthesis. So these are photosynthetic membranes here. And remember, the process of photosynthesis is simply using light from the sun plus carbon dioxide to make sugar. And the reason we do that is you are trying to store energy. All right. Oops. So just as a kind of quick basic review, these photosynthetic membranes that you find inside some prokaryotic cells, what they are able to do is use proteins that are embedded in these membranes, and they can take light energy and carbon dioxide. And since we can't store the energy of light, what we can do is convert that light energy into chemical energy in the form of sugar. And then that can be stored and used later to drive other cellular processes. As a matter of fact, later on, what you can do is you can trade that sugar in and turn it into ATP to drive cellular processes, okay? And that happens through what we call cellular respiration. So that's a preview of what's coming later, okay? All right. Let's see here. Okay, so that is enough about prokaryotes. Let's now move on to eukaryotic structure. All right. So, and a couple things I wanted to mention about eukaryotic structure before we get into all of this rigmarole here. All right. First of all, remember that we have a real big size difference between prokaryotes and eukaryotes. And that sets up an immediate problem for eukaryotes, okay? And it kind of explains why they have these membrane-bound organelles. And the issue has to do with something called surface area to volume ratio. Now, what I find is you may have heard this before and it probably didn't make any sense, but I think I have a way that's gonna make sense for you. I'm going to change the thickness of my pen. I'm gonna make this really thick. I want you to envision an M&M, and I want you to envision two different types of M&M. I want you to envision the M&M Mini. And then I want you to envision a regular M&M. Okay, M&M Minis, sometimes we use those in baking. Let me go back to my regular size here. All right, sometimes you use M&M Minis in baking, right? So this outer thick thing represents the candy coating, and the interior here is where the chocolate is. Candy coating, chocolate, okay? If we look at this little one, what you can see is that there's the proportion of candy coating to chocolate is pretty even Steven, right? Like, if you were to bite into this, you're going to get about as much candy coating as you do chocolate. And what that means is what's on the surface is almost equal to what's on the inside. The surface area to the volume is small. It's almost a 1-to-1, right? Maybe not quite, but you see where I'm going. Why is that important? Well, the reason that it's important is if you wanted things to be able to get into the cell, diffuse into the cell, or maybe you're using membrane transport to get them into the cell. But whatever the case may be, it's easier to transport things into the cell here and have them be distributed inside the guts of the cell. Now let's take a look at this bigger M&M, right? If you were to chomp down on this, you would get a lot more chocolate and less candy coating. Incidentally, that's why I think mini M&Ms are garbage and regular M&Ms are the way to go, because you get a lot more chocolate bang for your buck, right? So if you were to look here and try to figure out a ratio, this might be, let's see here. Let's say this is the inside, this one inside and this one is the outside. There might be 10 times the inside to 1 of the outside, right? There's just a whole lot more of the chocolate than there is of the candy coating. And so why is that a problem? Well, if you're trying to get things to diffuse to the interior of this, it's going to have a lot harder time, whatever molecule, if it's oxygen that we're trying to diffuse across this membrane and distribute it throughout the interior guts of this cell, it's going to have a lot harder time getting to the center and distributing, right? Because we have a lot farther distance to go. So how do eukaryotes overcome that? Well, the way that that happens has to do with compartmentalization. So I told you that eukaryotes, they have membrane-bound organelles, okay? So here's a nucleus, and maybe here's some other organelles. So eukaryotes are what we call compartmentalized. They have compartments. And so inside each of these compartments, we have smaller surface area to volume ratios where chemistry can occur. You can concentrate reactants, right? So you can concentrate chemical reactions, right? We don't have as much fluid in here that things have to dissolve in, that we can have things be transported in and out fairly easily. The other thing we can do is we can separate incompatible reactions. What do I mean by that? Compatible or ibble? It's ibble, I think. Ugh, hate spelling in front of people. Compatible reactions. Okay, so what do I mean by that? Well, what if you have an organelle here where you're making lipids for the cell membrane, but maybe you have an organelle here that's breaking them down, breaking down damaged lipids. You wouldn't want the enzymes responsible for breaking lipids down to be mixing with the enzyme responsible for manufacturing them, because then you might be breaking things down as quickly as you make them, right? And that would be very inefficient. So the fact about eukaryotes being big and having organelles, things I want you to know about that is when they're bigger, you have a greater surface area to volume ratio. And one way of overcoming that is through compartmentalization, where you can concentrate chemical reactions and speed them up, and you can separate incompatible reactions, okay? Now, just like with the eukaryotic cell, let's diagram our prokaryotic cell. And let me make sure I've got my notes here so I can make sure I'm diagramming everything I want you to see. So again, I'm going to draw an outline that is meant to imply for you a cell membrane. I'm drawing a single line. You guys know that it would be a bilayer, but we're not going to draw that, okay? So I'm going to label this as our membrane. All right. Now, many of you guys wouldn't already know that the nucleus is basically the big difference that you think of when you think of eukaryotes versus prokaryotes. I'm going to say nucleus. Okay. Now, the nucleus is surrounded by a membrane, but it's actually double membraned. Okay. And coming off of this double membrane, so as I was saying, the nucleus is double membraned, and you actually have pores in it here, where it's kind of riveted together, and these pores are little openings in the nuclear membrane. I'm also, you'll notice, kind of drawing an extension off here because we're going to use that in just a second as part of another organelle. Okay, so inside this nucleus, well, first of all, we have it surrounded not by a nuclear membrane, but by what we're going to call the nuclear envelope, because it is double membraned, okay? Now, what's contained inside the nucleus? Most of you would be able to tell me that inside the nucleus you have chromosomes, and if you're eukaryotic, they're usually linear. Okay, so I'm just going to draw a few in there. But you also would see inside the nucleus, sometimes it looks like there's another nucleus. There's often a darker region. And this is called the nucleolus. Okay, what is the job of the nucleolus? Oops, come on. It is where ribosomes are made, okay? So I'm going to put ribosynth. All right. Now, as part of this membrane system that's coming off of the nucleus, I'm going to draw a whole bunch of membranes here. What you will find is a series of membranes that are all kind of together coming off of the nucleus, and this is called the endoplasmic reticulum, and I am going to abbreviate that as ER. And you may remember from previous courses that you can divide the endoplasmic reticulum into two varieties, the smooth and the rough ER. The rough ER is dotted, okay? And the smooth ER is not. So what's the difference? The dots are actually little ribosomes. And the smooth ER lacks those ribosomes. So smooth ER, I'm going to write smooth here. It's going to be responsible for lipid synthesis. Come on, little guy, lipid synth. And of course, if you have ribosomes, the rough ER is going to be involved in protein synthesis. In addition to the ribosomes that are embedded and studded on the rough ER, you can also have free ribosomes that are hanging out inside the cell, okay? So I'm also gonna put that is a, actually let me try to make this as clean as we can. Let's, we don't need to draw an arrow here. Oops. Oops, okay. So these are ribosomes here. And I'm gonna just abbreviate RIB. Okay, so we have ribosomes on the endoplasmic reticulum, the rough ER, and we have free ribosomes out and about inside the cell, okay? Now, one thing that students often get confused are the ER and another set of membranes called the Golgi apparatus, okay? One way, if you were labeling a cell, that you could really tell is the ER is usually continuous with the nuclear envelope, right? Whereas the Golgi apparatus, you can, if you're looking at a cross section of a cell, it looks like it's kind of separate. So what is its job? So it's kind of works in conjunction with the ER, but what it does is it kind of packages, I'm going to put packaging. It packages things for export from the cell, packaging. It's like the Amazon distribution center for the cell. And so what it does is basically if you have, say, a hormone or something that's made in the ER, and these are actually kind of all connected behind the nucleus and stuff, what's going to happen is it's going to get packaged and maybe altered a little bit in the Golgi apparatus, and eventually it will make a little tiny vesicle, is what we call that, that will be moved out to the edge of the cell where it will fuse with the cell membrane and then be exported from the cell. So the Golgi apparatus is sort of the Amazon distribution center, the packaging center of the cell. All right, other things that I want you to know about, let's see here, lysosomes. I'm going to draw one of those in. So this is a lyso, and I'm going to some, so it's one word, but I'm trying to make room. If you see the word lyso, that means breaking, right? Breaking apart. And so you find lysosomes in animals, and they are typically associated with breaking down molecules that have kind of been worn out inside the cell. You can think of this as the recycling center, okay? All right, another thing that you might find is, let's see here, what else do I want to tell you guys? Peroxisomes, where are we at here? And just, okay, vacuoles, let's do vacuoles. The vacuole is a storage organelle. particularly in plants. All right. So lysosomes you find in animals, vacuoles you find in plants. Peroxisomes. Peroxisomes are also breakdown centers. They break things down. This is what we call a redox reaction center, peroxisome. All right, and I have all these, just in case you're wondering, down here for you guys, okay? So this is, we'll say it's for redox reactions, but a lot of those are often involved in breaking things down. These are also involved in detoxifying things in the cell. So we can put redox reactions or detox. I'll put detox here. We often use this for detoxification. All right, mitochondria. You guys have heard of mitochondria. You know they are the powerhouse of the cell. They are usually drawn, actually I made that probably too big. They are usually drawn to look like jelly beans that have sort of a lot of squiggles inside. And that's because mitochondria are double membraned. All right, we're going to talk about that in just a second. If you are a plant cell that photosynthesizes, you may also have another double membraned organelle that's got lots of stacks of membranes inside it, and that would be a chloroplast. That's responsible for photosynthesis. I'm going to put chlor here. So we've got mitochondria, chloroplast, peroxisomes, vacuoles. We also have cytoskeleton inside these guys, just like we did in the bacterial cell. Okay, so these provide structural support. I'm just going to put cytoskeleton. So that's structure. All right. If you are a plant cell, you may have an external structure that is made of a structural carbohydrate that is a cell wall. We talked about cellulose before. And so that would be for plants, right? So the cell wall of plants is very different than the cell wall of bacteria. This is made of cellulose. Bacterial cell walls are made of peptidoglycan, and they're very different structurally, okay? And then the last thing, and we'll talk about this a little later, is something called the extracellular matrix. What do I mean by that? Well, just like plants will sometimes secrete something around the outside of their cell membranes to provide structure and support, and that is the cell wall. Animal cells can also secrete things around the outside of the cell to provide structure and support. So I'm going to put structure here. And what they're secreting is usually carbohydrate and protein, and it kind of forms a rigid structure around the cell. A great example of extracellular matrix is bone. So you have bone cells that secrete the substance that will eventually become your bone, and the bone itself is what's the extracellular matrix, okay? So this is a lot. Let me show you where this is summarized for you. About 3 slides down, I've got for you a table that is also present in your textbook. And this is everything that I've gone over. Yes, I expect you to know this table because this should be review for the majority of you. All right. Okay, what else do I want you guys to know? Remember that what I just drew for you is a generalized eukaryotic cell, okay? An animal cell I have for you here. Notice it lacks a cell wall, but do you see here these, these fibers here and this gray stuff? This is meant to designate for you the extracellular matrix. So I want you to know that you've been taught that animal cells have these membranes, and they do, but they often secrete substances around the outside that provide additional support. Extracellular matrix is that stuff, okay? Other things that you might have in here. One thing I didn't talk about is centrioles. go ahead and look that one up because this lecture is getting long and these are just easily looked up functions, but everything else on here is stuff that we have talked about. Here is your animal cell. Notice that your animal cell has some cell walls around it to give it support and rigidity. Also notice there's some little tunnels. We're going to talk about these later in another chapter that allow these cells to have access to one another, okay? We'll talk a little bit about those at another time. Here you can see your chloroplasts. You can see mitochondria here. We're going to talk about those. I'm going to skip this. This lecture's getting long. So I am going to actually save this slide and this slide for a future lecture. So the last slide that you will be responsible for your exam is this one right here, okay? Hopefully all of this is review for you and this is helpful. Best of luck in your studying and I will talk to you soon. Bye-bye. Hey folks, just a quick correction. I was going back through my video and I realized I kept saying animal cell when clearly on this slide, I'm talking to you about a plant cell. So please recognize that I just was misspeaking and correct your brain accordingly. All right, folks, happy studying. Bye-bye.